Hello and welcome back to the Biochemistry for Health Sciences channel. In this video, we shall continue our discussion on the applications of enzyme kinetics, and we'll talk about how enzyme kinetics can help in the diagnosis of diseases. So, can enzymes help us diagnose diseases? The answer is yes. We measure the amount of enzyme or the concentration of enzyme routinely. So for example, the transaminases like ALT and AST are helpful for liver diseases. The enzyme alkaline phosphatase is routinely measured for liver and bone disorders. Enzymes such as amylase, lipase are used for pancreatitis. Chymotrypsin and trypsin in stool samples are used in cystic fibrosis and to determine pancreatic function. Creatine kinase and its isoenzyme is useful for heart attacks. LDH and its isoenzymes are for diagnosis of various disorders. And the enzyme renin is helpful for any problems at the adrenal gland, especially with aldosterone secretion. So if the concentration of the enzyme is increased, so for example, if alkaline phosphatase concentration is elevated, then the next thing we can do is we can detect using isoenzyme analysis where is this alkaline phosphatase coming from. Is it coming from the liver, or is it coming from bone, or is it coming from somewhere else in the body? So what's the rationale for measuring enzymes in body fluids such as blood and serum? So basically, if the cells were healthy in a normal person, these enzymes that are present in the cells, we would hardly see any of these enzymes in our bloodstream. Okay, very, very low levels of these enzymes in our bloodstream, because even a normal person, some cells are always leaky or cells are dying to a small extent, and we might find some basal level of enzymes in our bloodstream. However, when there is a disease, when there's a cell damage or the cells die, then these enzymes inside the cell, they are going to escape into the bloodstream. Now, if we take a sample of blood and analyze for the enzyme in the blood sample, we will see an elevation of the enzyme concentration. So how do we determine the concentration of enzyme in a body fluid such as serum? Serum is probably the most common body fluid that is used in a clinical laboratory. So we'll just basically focus on serum. So how can we measure the concentration of enzyme in serum? There are two ways that are commonly used. One is to actually measure the concentration of the protein. So for example, CKMB, we can actually measure the concentration of CKMB, and this is most often done by a technique called immunoassays. So here there is we need an antibody against creatine kinase MB, and if we have the antibody, we can perform an immunoassay to detect CKMB. We'll talk about immunoassays in future videos. So besides immunoassay or determining the actual concentration of the enzyme protein, there's a second way of measuring the concentration of an enzyme. Now, since enzymes catalyze reaction, we can actually measure the function of the enzyme, and this is called enzyme activity measurements. So we can measure in enzyme kinetics, we talk about Vmax. So we can actually measure the Vmax of the enzyme in the body fluid, because the Vmax is proportional to the concentration of enzyme, as we shall see in a few minutes. So while immunoassays will give you the actual concentration, so for example here we have nanograms per mil, because remember enzymes are found in very, very small amounts, so having nanograms per mil or picograms per mil is quite common. Enzyme activity, on the other hand, the most common unit is international unit per liter, IU per L, international unit per liter. 1 international unit is the amount of enzyme that will convert 1 micromole substrate in 1 minute under specified conditions, especially pH and temperature. So since this video is on enzyme kinetics, let's look at this part here, how we determine the Vmax and express it as international units per liter. So this is how we can determine the Vmax in international units per liter. So basically we take the body fluid, so let's say we take 5 microliter of serum, and then we add the assay reagent. So let's say this is 500 microliter of assay reagent, and in this assay reagent there is the substrate for the enzyme. So if this enzyme was alkaline phosphatase, then the substrate would be para-nitrophenyl phosphate that we talked about in an earlier video. Now the substrate concentration is present in excess, saturation amounts, okay, because Vmax can only be measured if the substrate is under saturated condition, is in excess. And when we do this, we monitor the absorbance at 405 nanometers, and then we determine what is the change in absorbance per minute. So if the absorbance change is 0.2 in 5 minutes, then 0.2 divided by 5, that will give us a delta A per minute of 0.04. So this delta A per minute can be converted into concentration of the product, para-nitrophenol, by dividing the delta A per minute by this extinction coefficient and the cuvette pad length. So if the cuvette pad length is 1 centimeter, then the amount of product para-nitrophenol per minute that is formed is basically the delta A per minute divided by the extinction coefficient, because the pad length is 1, so that's just multiplied by 1. We then multiply this delta A per minute divided by the extinction coefficient by the dilution factor. How much did the sample get diluted? So basically we had a total volume of 505 microliters. So that's 505 microliters, a total volume. The actual amount of sample serum was only 5 microliters. So this will give us a dilution factor. So that comes out to be 0.04, which is your delta A per minute, multiplied by 18.8 times 1. Dilution factor is 101, and this comes out to be 0.22 millimolar, because the units of this 18.8 is in millimolar. So 0.22 millimolar para-nitrophenol phenol is formed in a minute. So 0.22 millimolar para-nitrophenol per minute. So that's the amount of product that's formed per minute. So since 1 micromole of substrate gives you 1 micromole of product, this is basically the same 0.22 millimolar of substrate that has been consumed per minute. Okay, so this comes out to be 0.22 instead of millimolar, there will be millimole per liter. So this is the same as millimolar, and this is per minute. Now, since 1 international unit is 1 micromole of substrate consumed per minute, which is the same as micromole or product formed per minute. So we convert this millimole into micromole. We multiply this by 1,000. So this will be 0.22 times 1,000. That comes out to be 220 micromole per liter per minute. So since 1 micromole per minute is 1 international unit, so 220 micromole per minute per liter will be 220 international units per liter. So that's how we get this enzyme activity, the Vmax, as international units per liter. So these enzyme assays that are performed in the clinical lab, they are always done under saturating substrate concentrations so that we are finding Vmax. So this is a Vmax. These are all Vmax that we are finding. Okay, so you can see that the Vmax is proportional to the concentration of enzyme. Okay, so by measuring Vmax, we are actually measuring the concentration of enzyme, and we express the concentration of enzyme as international units per liter. So this is what you commonly see when you get a clinical lab report. The enzyme units are in international units per liter, okay, IU per liter. So if we see an increase ALT, so you can see, for example, in this report, the normal range for the healthy range for ALT is less than 71 international units per liter. Okay, so the range is 15 to 71, so less than 71. So as long as the person's value is less than 71, we do not suspect any problems with the liver. But if the value increases, let's say to 500 international units per liter, then we suspect that there may be some problem. The liver cells may be undergoing some sort of damage or maybe even dying. So besides actually measuring the concentration of the enzyme in the body fluid, there is another way by which enzymes can be very helpful in the diagnosis as well as monitoring of diseases. So for example, let's say the alkaline phosphatase for this person, the normal range is less than 154, but let's say we had a value of 300 in this patient. So that means there is some enzyme that is coming out from cells in the person that are either being damaged or that are dying. Now the question is, what cells are being damaged or dying? Are they liver cells, or are they cells in the bone, or are they cells maybe in the intestinal part of the body, the intestine, the GI tract? So what exactly is happening in this person? And to answer the question, where is these high levels of alkaline phosphatase coming from, we do what we call an isoenzyme analysis. Okay, so by analyzing isoenzymes, we can tell where exactly is the enzyme coming from. Is it coming from liver, bone, or somewhere else? So before we talk about isoenzymes, a few definitions will be good to review at this point. Isoforms. What are isoforms? So isoforms are proteins that are quite similar to each other in terms of structure and perform similar biological roles. So example, beta-actin is an isoform of gamma-actin. Okay, so beta-actin and gamma-actin are examples of isoforms of actin. Now, isoforms may be made from separate genes. They may come from separate genes, or they may come from a single gene. The same gene codes for the different isoforms, but the reason why the isoforms exist in this case would be because there is differential mRNA splicing. So remember, it's how we connect the exons together. Okay, so it's a differential mRNA splicing. So although isoforms are quite similar, so this is important, quite, not totally similar, they do have different amino acid composition and sequence, therefore resulting in differences in structure or structural variations. Isoenzymes are simply isoforms of enzymes. They catalyze the same reaction, but they have slightly different sequences and structures. So let's look at some examples of isoenzymes. So the first example is the isoenzymes or the isoforms of the enzyme creatine kinase. Now, there are two types of subunits for creatine kinase. This enzyme is made up of two types of subunits. There is a M and a B, but by various combinations of M and B, you can see we can get three isoforms or isoenzymes of creatine kinase. If two M's combine together, they will give us one type of isoform, which we'll call CKMM. If an M combined with a B, that will give us a second isoform or isoenzyme that will be called CKMB. And then in CKBB, this is a B unit and this is also a B unit. So just from the various combinations, we can get three isoforms or isoenzymes of creatine kinase. Why does our body have these various isoforms of creatine kinase, where all of them do the same reaction? They catalyze the same reaction. MM catalyzes this reaction, MB catalyzes this reaction, BB catalyzes this reaction. Well, we believe that the different isoforms, for example, the MM is more predominant in skeletal muscle. Probably it works better because the oxygen levels in skeletal muscle fluctuates quite a bit. On the other hand, in the heart, where we have predominantly MB, there is constant oxygen levels, so maybe the MB works better under constant oxygen levels, while MM is more effective under fluctuating oxygen levels. So the different isoforms of a protein or different isoenzymes of an enzyme are made so that they work better in their respective tissues. Here's another important example clinically of an enzyme that has various isoforms, and that is lactate dehydrogenase. Now, lactate dehydrogenase is a tetramer. It has 4 subunits. And there are two types of subunits available, the M and the H. So in the isoenzyme called LDH1, all 4 subunits are H, so we call it H4. In LDH2, 3 of them are H, 1 of them is M, and so on. LDH3 has H2M2, LDH4 has 1 H and 3 M units, and LDH5, all the 4 subunits are muscle, the M type. So you can see the different forms, the different isoenzymes of LDH are more predominant in certain tissues. Like LDH1 is more common in heart and red blood cells, while LDH5 is more predominant in skeletal muscles and liver. And it's believed that LDH1 is much better in catalyzing lactate to pyruvate, while LDH5 is much better in catalyzing the reaction in this direction, the reverse direction, pyruvate to lactate. Another example of a clinically important enzyme that exists in various isoenzymes or isoforms is alkaline phosphatase. So once again, there are different kinds of alkaline phosphatase, part of it due to variations in the primary structure, and then part of it because alkaline phosphatase is a glycoprotein, so the actual carbohydrate content of these different isoforms varies from one to another. So we have alkaline phosphatase that comes from the placenta. We call this placental alkaline phosphatase, PLALP, also called the Regan isoenzyme. We have the intestinal alkaline phosphatase called IALP, which is very heat stable. We have the liver bone kidney alkaline phosphatase, also called LBKALP. This is very sensitive to heat. And then we have the germline or GCALP, germ cell alkaline phosphatase. So 1, 2, 3, 4 possible types of alkaline phosphatase. So because of their slight structural variations, these isoenzymes can be separated quite well by a technique called electrophoresis, where we apply a sample on a gel. In the clinical lab, the gel that we use most often is agarose. Of course, in research and more specialized labs, they also use polyacrylamide or PAGE. But agarose is commonly used in most hospital labs. So we basically apply the serum sample here, and then we turn on the electrical current, and the protein will start migrating. So originally, you will have only one band, and then eventually they will separate because their net charge is different from one isoenzyme to another. So they will separate as into the various isoforms. Same thing here, lactate dehydrogenase is applied here, and it will separate into its bands. And here we see alkaline phosphatase, and they will migrate differently and therefore separate on the gel differently. Now, after running the electrophoresis, of course, we're not going to see anything on the gel. Okay, so the gel is basically blank. There's nothing on the gel you can see because proteins do not have a color. So your alkaline phosphatase or your creatine kinase and so on, they have no color, so you're not going to see any bands. But then what we do is we take this gel and we incubate in the enzyme reagent. So for example, if this was a ALP analysis, alkaline phosphate analysis, we would use the ALP reagent. We will incubate the gel into this ALP reagent. So what happens is, if there is, let's say, an alkaline phosphatase band somewhere here, there was alkaline phosphatase here, this enzyme will convert the substrate. So here the substrate that is used is a specialized substrate, 5-bromo-4-chloro-3-indole phosphate, and the phosphate is removed. That's what alkaline phosphatase does. It removes the phosphates from organic compounds, and we get an indoxyl intermediate formed here, and this intermediate in the presence of nitro-blue tetrazolium, NBT, it will react to give us a product, and it gives us another product where the NBT becomes insoluble. This is blue in color. So this insoluble NBT diformation, this product is blue in color, and because it's insoluble, it precipitates in the gel. And what we see is a blue band. Wherever the alkaline phosphatase is present, the reaction is being catalyzed, and you see blue bands. So in this case, we see a very light intestinal band and a reasonably strong band from the bone and a band from the liver if we had all the three isoenzymes present. Electrophoresis was traditionally done manually. However, today we have some very good instruments that do all the steps, the actual electrophoresis as well as the staining and so on, automated. And here's an example of a clinical gel electrophoresis system. So this instrument doesn't only just run the electrophoresis and then stain the bands, but it also then scans the stained bands, and it gives you this profile, this scan. Okay, so there's a beam of light that will that will run through all the bands, and it will tell you the intensity of the bands, how intense the bands are, and then it will give you a scan. And this scan, from this scan, these profiles, we can calculate what percentage is LDH5, for example, what percentage is LDH4, what percentage is LDH3, 2, 1, and so on. So this is called a densitometric scan of the bands in the gel. So for LDH, the incubation mixture is this, because LDH will catalyze this reaction, so the substrate will be lactate and NAD. And if the enzyme is present, like for example here, if the enzyme is present in the gel, it will catalyze this reaction, and what we get is pyruvate and NADH, and this NADH will then react with NBD, which is in the incubation reagent, as well as PMS, which is present in the reagent, to give you NAD, and you get this blue precipitate, this blue formosine precipitate. So wherever the band is present, you will see this blue precipitate appearing on the gel. And then the instrument will scan the intensity of the band and give you a profile. So here's an example. So you see a normal profile here. So the sample is applied somewhere on this side, somewhere here on the gel, and then it's run in that instrument. And you see a normal profile, hardly any intestinal band, relatively visible bone band, and then the one from the liver. And here in patient number 3 here, you see a very strong liver band, showing the person has liver cells that are being damaged or destroyed, some sort of liver disease. And in this person here, you see a very strong band that is migrating similar to the bone. So this clearly shows that some cells in the bone are being damaged or are being destroyed, dying. And they're releasing the alkaline phosphatase into the serum, into the blood. And that explains this very strong band here. Okay, so just to conclude, can enzymes help diagnose diseases? Yes, we measure the enzyme activity. Usually we express very commonly, the units are international units per liter, and you see that in your report, in your clinical lab report. That's what this means, IU per L, international unit per liter. And if necessary, if we want to know exactly where the enzyme is coming from, then we can also do an isoenzyme analysis. So yes, enzymes are very useful for the diagnosis as well as the monitoring of diseases. That is all for now. Until the next video, goodbye and have a great day.